Dr. Mark Schwartz, CEO of Galena Biopharma. Before Galena, I was CEO of several venture capital-backed companies. The last company was an autoimmune company working in the area of T-cell tolerance called Bayhill Therapeutics. So there's a number of approaches in the immuno-oncology space. Certainly the ones that have garnered the most attention and have been most successful have been the checkpoint inhibitors, Keytruda and Opdivo. They are designed to take the brakes off the immune system, so they don't really directly change or impact a T cell directly as much as they just allow the immune system to let the T cell do the job that it was designed to do. I think there's a number of other approaches that are in the clinic and being worked on that are really centered more on directly affecting the T cell, such as T cell stimulators or autologous treatments where you're taking a T cell out of the body and treating them in some manner to generate a certain antigen specificity. Galena is working on cancer vaccines that are designed to increase the number of antigen or target-specific T cells, in particular Nuvax, which is targeting HER2-expressing tumors, in particular breast cancer. So cancer vaccines have been worked on for a number of years and admittedly have not had the kind of success that I think everybody had hoped for. I think one of the challenges is not so much the ability to generate a potent immune response. A lot of different approaches and compounds and drugs can generate an immune response. The challenge is what setting are you putting those drugs in? What's the specific stage of the tumor? Is it a high disease burden or a low disease burden? And what's the setting that you're asking the immune system to do the work that you're asking it to do? So many of the previous vaccines were in the metastatic setting or in late stage tumor settings where the tumor had already built in its immune escape mechanisms. We, Galena, as an example, we're working in the adjuvant setting, the minimum residual disease setting, where we know there's only a few cancer cells, micrometastases, and it's a much more potent framework by which the immune system has to do its job, which is seeking out and destroying tumor cells. So we use a peptide vaccine, a peptide which is a piece of the HER2 protein, to immunize the patient. When we immunize the patient, we generate a significant amount of clinical data that show we generate a very potent T cell response. Upwards of about 2% of the total T cell population is specific for Nuvax or specific for HER2 expressing cells. In this case, HER2 expressing cancer cells. Again, going back to my previous comment, the real advantage is not so much the fact that we can generate this potent immune response, but the setting in which we're generating it. So we are now treating women who've gone through the standard of care. They are disease-free. They have no detectable cancer and healthy immune systems, and that's an important point. They have healthy immune systems. They are now told to go home and watch and wait. If they're HER2 1+, 2+, so they overexpress HER2, but not at the highest levels, they don't have any HER2-specific treatment. When they are then immunized with Nuvax, their immune system expands significantly. The T cell population specific for HER2 expressing cells, those T cells can then go seek out these single cold cancer cells. Cancer cells are hidden away in the body and quiescent, or micrometastases, very, very tiny microtumors that haven't yet taken on the full characteristics of a tumor, have not yet learned to avoid the immune system. The immune system can seek those out and destroy them before they come back as a secondary recurrence. And I would add at this point, when there is a secondary recurrence, when a woman who has been initially rendered disease-free and has a recurrence, those recurrences have a very, very poor prognosis. Oftentimes they're fatal. Well, there's a number of immunological markers that we look at. We do what's called a DTH test, delayed hypersensitivity test. We've actually drawn blood samples and measured the number of CD8 T cells and the specificity of those cells, and that data has been highly published. There is no question that we are not generating a very potent and a very specific immune response. The real question that clinical trials were designed to answer is the generation of that potent immune response in the setting we've chosen, the adjuvant setting, going to produce a clinical response of disease-free survival. So the data from our phase 2 trial indicated that Nuvax, which again is a small peptide off the extracellular domain of the HER2 protein, the data from our phase 2 trial indicated that Nuvax looks to be effective in reducing recurrence in women who are HER2 1+, 2+. And while they are called clinically HER2 negative, i.e. they're not suitable, they're not indicated for Herceptin, they do overexpress HER2. And since there is no HER2-directed therapy for these women, it is an unmet medical need. There's very, very few people working in this area. There's no late-stage compounds in development. This is a need that physicians do have to treat these women. So we decided to go after the HER2 1+, 2+ patient population based both on the success in the phase 2 clinical trial as well as the unmet medical need in the physician's office. We have about 1 year of cash left in the bank right now, and so that should get us through some critical milestones coming up, in particular our interim analysis in the phase 3 PRESENT trial. We expect right now to be in the second quarter of this year. We have a few key strategic partnerships. We have a license agreement with Dr. Reddy's in India and Teva in Israel. We do continue to have discussions with potential partners, but we want to make sure that we preserve our shareholder value in the long term, so we will take those as they come.